Hello students, in this video we're going to talk about some of the basics of genetics starting with Mendelian genetics. So first to talk about Mendelian genetics we need to talk about who Mendel even was. So Mendel is oftentimes considered the father of genetics, and this was due to the work that he did in the 1800s with the pea plants. So to start off, we're going to go into some review about what genes actually are. Once we've gone through that, we're going to go into the experiments that Mendel actually performed and how he came up with his law of segregation and his law of independent assortment. In addition to this, we're going to talk about probability and what role it plays in genetics. Not covered in this video will be some extensions to Mendel's model, because really, just like everything that we begin with in science, it's an oversimplification. There are more complex things going on here than what we are actually just looking at with Mendel. So in the next video, we'll talk about some of those extensions. So first of all, what are genes? So genes are specific places on chromosomes that are going to code for a specific trait. These genes exist in pairs because you have sets of homologous chromosomes that come in pairs. So here I have a pair of homologous chromosomes. The purple stripe here represents a specific gene on these two chromosomes, and so this would be one allele of the gene, this would be the other allele of the gene, so you'd have two alleles. Now it's possible that they're going to be the same allele, in which case you'd be homozygous, so here we are homozygous for the purple allele, or you could have two different versions of the gene, in which case you're going to have one allele that is slightly different than the other. That difference comes in the actual DNA that makes it up. So this individual would be heterozygous because they have a white allele and a purple allele, and this individual would be homozygous because they have two white alleles. Ultimately, we will find that the genes, or the genotype, is determined by the particular DNA that is present within the chromosome. So it's the sequence of base pairs in the DNA that controls the genotype, and ultimately the DNA you have controls your traits because the DNA encodes for proteins. So it's the presence of a protein that leads to the phenotype the organism it actually exhibits. Now, what DNA and protein is present in your cell is not the end-all of your phenotype. There are environmental factors playing a role as well, but to begin, we're going to just look at DNA as being what's responsible for your particular phenotype by controlling what proteins are actually present. So in our first example, we're going to talk about Mendel's experiments with pea plants as it pertains to the flower color that they had. So you can have two different types of flowers. You can have a purple flower or a white flower. You get white flowers when you have two copies of the white allele, and you get purple flowers when you have two copies of the purple allele, or you have a purple allele and a white allele. So we call the purple phenotype here the dominant phenotype, and we call the white phenotype here the recessive phenotype, because you only need one copy of the purple allele to actually see that particular phenotype. Alright, so let's talk about Mendel's experiments. In his experiments, he would take true breeding populations of his traits. So he would have a true breeding white flowered pea plant, which means that every time he collected offspring from these flowers, they would always have always be white flowered offspring. And he had true breeding purple flowered pea plants as well, so that would always produce the purple allele. This is the P generation. He would cross these two these two true breeding plants, and he would get the F1 generation. Now, he found in his F1 generation that the dominant phenotype was the only phenotype that he would observe. However, when you would take two individuals from the F1 or take an individual and self-fertilize in the F1 generation, he would end up mostly with the dominant phenotype, so with purple flowers, but about 25% of the time he would end up with the recessive phenotype, having white flowers. So what's going on here in this F2 generation? Well, to illustrate this, we're going to use something known as a Punnett square. So that's what's drawn over here. What's going to go on top here are the different alleles that I can get from one parent, and then over on the sides here, I'm going to put the different alleles that I get from the other parent. And what alleles I can get is dependent on what alleles are present here. So you can see for the white plant, I have two chromosomes with the white alleles on them. So this represents a gamete. This gamete would get this allele, this gamete would get this allele, and then this would be the other parent's gametes. This one would get a purple allele, and this one would get a purple allele as well. We're going to use letters now to illustrate the actual genotypes that they have. So we have a big P for the dominant allele and a little p for the recessive allele. Now we just fill out these Punnett squares, so this big P is going to go in this square and this square. This big P here is going to go in the top right and the bottom right. And then this little p over here on the top left, it's going to go in the two top squares, and this little p is going to go in the two bottom squares. And we end up ultimately with all of the plants having the same genotype and phenotype. So the genotype are the two big P, little p, and the phenotype is what's actually being exhibited. So that's the purple flower. Now let's look at the cross that produces the F2 generation. So now we have individuals that have a big P and a little p present here. We'll put those up on the Punnett square. And if you see the letters, we have big P, little p, big P, little p. We're going to fill this out like we did before. So this is going to get two big P's, this big P and this big P. This one here and this one here are going to have a big P and a little p. This big P is going here, this little p is going here. This big P is going down here, and this little p is going down here. And then the last square is going to get two little p's. And so we end up with the anyone that has a big P in it being the purple flower color, while anyone that has the little p's in it being the white flower color. All right, so this brings us to Mendel's two laws. So the first law is the law of segregation. Simply put, the law of segregation is during the process of gamete formation, any alleles that are present in the organism is going to separate into two separate gametes. So what we're saying here is that for this heterozygous individual, this allele is going to separate into one gamete, and this allele is going to separate into the other gamete. And what's going on here relates to meiosis. So remember, during meiosis, you had the homologous pairs line up during meiosis 1, and then they would separate during the anaphase 1 of the cycle. And so the alleles are separating because they are actually a component of the chromosomes that are separating during meiosis 1. Now, the law of independent assortment relates to meiosis as well. And what it is saying is that how one set of allele pairs lines up and separates does not affect how another set of allele pairs line up and separate. So you can have this orientation here where the white and the yellow are on the same side and the purple and the green are on the same side, and then when they separate they're going to go into their own alleles. Or you could have this set up over here. So the white is on the left and the green is also on the left instead of the yellow, and purple is on the right and yellow is on the right. So you can end up with 4 different gamete possibilities in this scenario. You can have white with yellow, you can have purple with green, you could have white with green, and you have purple with yellow. And that's going to be the basis for a dihybrid cross. So Mendel did more experiments than just looking at flower color. Another trait that he would look at was seed color, and then another one was seed shape. And what he found was that the dominant phenotype for seed color was yellow, and the recessive trait was green for seed color. But when it came to shape, the dominant phenotype was round, and the recessive phenotype was wrinkled. And so he would have a true breeding population of round yellow seeds and a true breeding population of green wrinkled seeds. And he would perform a cross. So that cross ultimately would give you a round seed that was yellow, but that is heterozygous for both of these traits. So big R, big Y, little y. Now, when he did the dihybrid cross, we're going to need more squares than our 4 square Punnett square. You have to take into consideration each allele pair that can line up. So this big R can go with the big Y, and the little r here can go with the little y. So that's what is represented here. We have the big R and the big Y, and we have the little r and the little y over here. And then we have the same thing up here on top because we're crossing, doing a self-cross with this. In addition to this, you can have the big R go with the little y, and the little r go with the big Y. And so that's what is represented by these two middle crosses here. So this big R can go with the big Y, this big R can go with the little y, the big R can go with the little r can go with the little big Y, and the little r can go with the little y. But what you don't get is a big R and a little r, because these are in allele pairs and they separated per the law of segregation. So now we fill out this Punnett square just like we filled out the other Punnett square, except that we have to do more of them. So this is going to get big R, big Y all the way across here, big R, little y all the way across here, little r, big Y all the way across here, and little r, little y all the way across here. And then we do the same thing going down, and that gives us all of our genotypes. So these are the genotypes that we get. The phenotypes that we get, how they actually appear, look like this. So you see that we end up with mostly yellow and mostly round seeds. Most of them are going to be yellow and round. If you count these, you see 1, 2, 3, 4, 5, 6, 7, 8, 9 are yellow and round. 3 of them are going to be green and round. 1, 2, 3. 3 of them are going to be yellow and wrinkled. 1, 2, 3. And 1 of them has both of the recessive phenotypes, the wrinkled and the green. Now, we came to this by looking at a dihybrid cross, but we can do the same thing using the laws of probability. So there's 2 rules that are pertinent to genetics, and that's the rule of addition and the rule of multiplication. The rule of addition says that when 2 outcomes are independent, so they can't both happen, you can get the probability of either of those happening by adding them together. So what does this look like? So for a monohybrid cross, if I wanted to know what the probability was of getting a dominant phenotype, I would have to look at the probability of getting a homozygous dominant genotype and a heterozygous dominant genotype. So big A, that has a 1/4 chance of happening, and the big A, little a has a 1/2 or 2/4 chance of happening. So the chances of me getting a dominant phenotype is going to be 1/2 plus 1/4, or 2/4 plus 1/4, so 3/4. Now, the rule of multiplication is saying when two things are not independent, so that they both can happen. So in this case, we're going to be looking at what is the probability that I get both the recessive phenotype for the A trait when two parents that are heterozygous are crossed, and what the probability of getting both getting the recessive B phenotype as well. So I have a 1/4 chance here and a 1/4 chance here, meaning that 1/4 times 1/4 is 1/16. I have 1/16 chance of getting little a, little b, when I have parents that are big A, big B, little b, and cross them. If you do this and look at what we got for the mono or the dihybrid cross, so the chances of me having a dominant phenotype over here is 3/4. Chances of me having a dominant phenotype over here is 3/4. So 3/4 times 3/4 is 9/16. Chances of me having dominant A but recessive B, so 3/4 times 1/4 is 3/16. Chances of me having a recessive A but a dominant B, so 1/4 times 3/4 is 3/16. And I already mentioned 1/4 recessive and 1/4 recessive leads to 1/16. So that's where the 9 to 3 to 3 to 1 ratio comes from.